So this is what we're looking at as you learned in your module for the tongue slide. So here we go. So we've got these gustatory sensations, and these allow us to detect foods, poisons, minerals, etc. And we learned in there that our sense of taste also involves our sense of smell. But when we taste food, the receptors are located in structures on the tongue that are called papillae, nipple-shaped mounds. These bumps that you have on your tongue, you also have these papillae on your soft palate and also on your throat. So if we look at those papillae, we're going to focus on this type right here that are called valley papillae. So here is a blow up of that bump, and notice that there are these crevices right here. So when you eat your food, your saliva mixes the food and breaks it down, and that food goes into these cracks that you see right here. And when it goes into these cracks, here's another view of them right here, we're going to see along the side of these cracks these structures that are called taste buds. And taste buds are just collections of many of these epithelial cells. And they contain the receptors for the sense of taste. So we have these supporting cells right here, and then we also have these, in beige are the supporting cells, and then we have these gustatory cells. And notice that these gustatory cells have these cilia, stereocilia, microvilli at the end, that are going to bind to the chemicals in the food. And what do you see coming out right here? What are these represent over here? These are axons that are all going to come out and go to the regions of the brain so that we can sense that. So, and then here are the basal cells right here, the stem cells that are going to regenerate and give us more of these gustatory cells. So this entire structure is called a taste bud, and that's what we're going to need to be able to identify in our lab. And so what that looks like, if we look over here, this nice image that was provided here by Raphael, you can see these taste buds along the edge. But let's go ahead and put also on here, let's also look over here, and when you come to Open Lab Friday, Saturday, and Sunday, this is what you are going to see. Sorry. So this is what we're going to see. One second. Sorry. Let's try this one more time. It needs to reboot. Nice. Okay, so what we are looking at right here, let me turn off one light so that we can see just a little bit better. If we look up here, what we see, open that up, is a slide of the tongue. And so we're going to have to look around to identify that region there that has the papillae. So all of this that we're seeing right here, this is just a shot of the tongue. That's all skeletal muscle at low power. And then you can see here identify the type of tissue that we see right here at the top. Identify the type of tissue. Stratified, squamous, keratinized, or not. The only part where we have keratinized is the skin. And then we move along, and then can you see the papillae there? The nipple-shaped mound? And this is where we're going to look in the crevices. This is where we're going to see all of our taste buds along the edges there. So that's at low power. Here it is at higher power. Right there. And then now I'm going to put it one higher power. Let's put that in the center. And then let's go one higher power to right there. Those beautiful structures. Can you guys see that taste bud right there? coming out. There's the little pore, so the food is going to go down here. It's going to bind to receptors on there. So you just have to be able to identify for your practical exam, these taste buds that are here along the edges. Okay, so everybody good on that? We'll probably have one image, most likely at low power, that you see like this, where you can see the villate papillae, and then over here we'll probably have a zoom after that that shows you nicely all of those various taste buds located. There are about 10 of them all lined up, one next to the other. Okay, everybody good? Okay, that's the tongue. The next slide that we have to look at on page 2 of your lab handout is the olfactory epithelium. So for the olfactory epithelium, let's switch over here back to our PowerPoint slide. Put on one light. And let's look here. Yeah, here we go. At this one, which is just showing you in the olfactory epithelium. So in our olfactory, there are about 1,000 different types of smell receptors. Each one binds to a particular part of an odor molecule. And so those are located at the very top of the nose. That olfactory epithelium is located at the very top, where we see there in blue. Does everybody recognize right here? This is going to be, this is what it's going to look like. There's our medial septum, our septum, nasal septum, made up of the vomer and the perpendicular plate of the ethmoid. And then there at the very top on either side is where we have our olfactory epithelium. And so when we look at it, it's just modified pseudostratified columnar tissue, and you can see at the end these hairs that it has at the end, and those hairs have the receptors that will bind to all of the chemicals in the air that you are smelling. Then you can see over here that there's also mucus. Notice that they also have those basal cells, those stem cells, the ones that we were talking about actually those were up in the olfactory bulb, but it has those as well. So this is what it's going to look like. You hopefully recognize the pseudostratified epithelium, and mixed in there are going to be several types of cells, and we just have to recognize it as olfactory epithelium. So when we put the slide on for that, what it's going to look like is let's put this over here. What it's going to look like at low power is going to be like this. So this is at low power. So I see all kinds of things going on there. I see, this, I'm going to see some hyaline cartilage in there. And what I want to do is I want to go to the very end right here where it's going to fork, and I'm going to focus in on this area right here. So this is hyaline. Hopefully you recognize this as hyaline cartilage with the lacunae and the chondrocytes in there. And this right here, both of these ends right here, at this end and is where we're going to see that olfactory epithelium. So that's at low power. Let's put it at medium power. Just a second. Focus there. At medium power, right here. So we're going to focus it in that region right there. And then at higher power, we can see just a little bit better that pseudostratified epithelial tissue. You can just barely see that on there. Can everybody see that right there? That pseudostratified. Let me get a little bit better region. Tissue. That's a better picture right there. There we go. You can even see there's some mucus and some things getting caught up there that were trapped there along the way in there. So that's our olfactory epithelium. Okay, so that's on page 2 of our outline. Now let's turn to page 3 of our outline, and this is where we start to look at the eye model. The first structures on the eye model are just the structures that we can see on the outside of the eye. So we're going to escape out of here, and we're going to come to this slide here. These are what we call the accessory structures of the eye. And so in these accessory structures of the eye, we're going to see the palpebrae. The palpebrae is just the fancy name that we use for the eyelid. We also need to identify this area right here towards the medial end. This would be the medial commissure. And we also have to recognize right here that the corners of the eye, where the eyelids come together, that is called the lateral commissure. And we also need to identify this thin membrane that's here on the top of the eye, and actually it lines the inside of the eyelids, and that is called the conjunctiva. You have probably heard of conjunctivitis before, or pink eye. That's where you infect that membrane. This is what it looks like. It's just a transparent membrane, and then can you see it lining the inside there of that eyelid? Here you can see a better view. Here's the conjunctiva right there. Can you see it wrapping around and lining right there the inside of the eyelids? And where the inside of the eyelids. So that is the conjunctiva. You also have to be able to recognize the lacrimal gland. This is the lacrimal gland on the model that we have in lab. This structure at the superolateral part of the eye, secreting tears. And then you also have to be able to recognize the lacrimal puncta. So the lacrimal puncta is just this little hole that we see right here, where the tears that are made up here in the lacrimal gland go across the surface of the eye, and they drain into the lacrimal puncta, and notice that they drain into the nose. That is why when you cry, you always need a Kleenex when you cry, and that's because you literally, tears literally go into your nose and loosen up the mucus, and that's why you need that Kleenex whenever you cry. You'll be crying tears of joy on Wednesday, and because you guys are going to do great. Yes, sir.
So it comes out of the eye too, right?
Yeah, it can go overflow on the eyelid. We actually, it's really neat if you ever have a chance to just Google about types of tears. There's different types of tears that you secrete at different points, but got to move on. All right, so then we also have the muscles of the eye. So the muscles of the eye that we have to be able to identify are the superior oblique. Notice that the superior oblique goes through this pulley, this trochlea that we identified earlier when we were talking about the cranial nerves. Then after that, we're going to have this one on the bottom that goes in an oblique direction, and then the other ones are easy after that. So we're going to have the lateral rectus muscle, the medial rectus, and so on. So if we look at our models over here, let's go ahead and look at our models. I'll put this on the camera. We'll probably have one that looks like this. Can you guys pass this back to Raphael? Thank you, Raphael. I just don't want you to lose that. If we look over here to our camera, what we are going to see is that we have to be able to recognize, let's see if we can put it this way, which is our medial and which is our lateral. So the side where the trochlea is located, that's going to be the medial part of the eye. So knowing that, I then know that this muscle on this side is the medial rectus muscle, and the one on this side is the lateral rectus muscle. This one must be the superior rectus, and this one here on the bottom is the inferior rectus muscle. So on this particular model that we have right here, the only thing that we can identify are those muscles right there. We do not see the superior oblique or inferior oblique. To see that, we would have to ask that to you over here on these other models that we have. So here's a nice example of this is nice. Sorry, we didn't have this one before. Okay, on this model right here, you should be able to see a little bit better. Right there. It's kind of weird, I'm sorry. Let's put this where you don't see the camera, the laptop there. Okay, so if we look at this right here, in this region, we see over here that trochlea, right? So we know that this muscle over here behind that trochlea is going to be which one? Superior oblique. And under all of this, that's where we're gonna find our inferior oblique. Which would this one be right here? Actually, this is another, is the superior rectus, and we have one other one located under there, and that is called, you guys have to identify the superior, yeah, the levator palpebrae superioris, and this is the one that is gonna voluntarily open and close the eyelid. That's the one that opens and closes the eyelid, and it's called the levator palpebrae superioris, and that's actually this one up here. The other one, the superior rectus, let's look at it here from the side, sorry, right here. See, this one is going to the eyelid, so this is the levator palpebrae superioris, and this other one right here underneath it, see that? That would be the superior rectus muscle. Okay, so identify the muscle. Lateral rectus, identify the muscle. Levator palpebrae superioris, identify the muscle under that. Superior rectus. And what's this one over here in the corner connected to this trochlea, that pulley there? Superior oblique. Very good. And then you can get the other ones. All right, and then after that, now we're going to start looking at the various layers of the eye. So let's, again, just, you can look at these, show you each of the layers of the eye. So for the layers of the eye, we're gonna divide the eye up into three major layers. What we're gonna call the fibrous tunic, the vascular tunic, and the neural tunic. Fibrous tunic is shown here in blue. Remember on the module, that included this structure right here, which is what? Cornea. And then from here all the way to the back, the white of the eye, the sclera. And then in the vascular tunic, it included three structures. This structure, which is the colored part of the eye, the iris. This structure right here, the ciliary body. And then this structure, which goes all the way here to the back right here, the choroid of the eye. And then the last layer is going to be the neural tunic, and that includes the retina of the eye. So let's look at our model, and let's identify those structures here in our model. So if I point over here to this region, right here, identify this structure right here. The colored part of the eye, iris of the eye. What's the hole in the middle of the iris called? That's called the pupil of the eye. It's just a space, it's just a hole. What about what's all of this white stuff on the outside? That would be the sclera. I don't have a cornea on the top of this one. I'm not sure where the cornea is off the model. If I take this off, this brown structure right here would be the choroid of the eye. And then if I take this off right here, we can see from a side view, right here, this is the sclera of the eye in white. Right here. Let's put that a little bit closer. This is the sclera of the eye right here. This right here represents the ciliary body. This represents the lens of the eye. And then here, lining the inside in beige, is the retina of the eye. This purple dot that we see right here is what we're going to call the fovea centralis, and it includes the macula lutea. This spot right here, where we see the optic nerve going out, that we're going to call the blind spot, or also called the optic disc. So again, looking in beige, all of this in beige is the retina. This purple spot Fovea centralis or macula lutea, this spot, optic disc, this structure right here, optic nerve, and then if I come over here, can everyone see those white structures right there? Those are called suspensory ligaments. They're going to attach to the lens of the eye, and they're going to shape the lens of the eye. Your lens always changes shape when you're trying to focus things. Like right now, maybe you're looking down at your sheet, and then you're looking up at the screen. So every time you look down, your lens focuses, and when you look up the screen, it focuses again. So those are going to pull on there, those white lines that you see there. This on the inside, this is the iris of the eye. Iris of the eye, suspensory ligaments, and then this structure that kind of like you see some scalloped structures right there. This is all called the oro serrata. And that just shows you where the retina ends and that ciliary body begins. This is just the end, the oro serrata. Those are the structures that you need to know on the eye. Everybody okay on those? Let's stop for a second.